Yeah, welcome everybody for our fourth annual in vitro software user meeting. Thanks for coming here, and also everybody that joined us online for today's session. We already had a great workshop around VivoQuant scripting yesterday, which many of you attended. That I heard was very successful and was well received. And today we wanted to present to you a little bit of our work that we have done over the last year, as well as give you an outlook on what we as the software group at Invicro are focusing on, and give a couple of our users also a chance to present on their work, which very likely have similar problems that you have and used Invicro solutions to solve them. And I also hope that we have a lively discussion about things. So please ask questions. This is also more like a workshop where we can also go into more details of things that are of interest to you. So please feel free to come up with questions. We are also, that's not working yet. Yeah, we are also celebrating actually today the 10-year anniversary of Invicro. To the day, 10 years ago was our incorporation of the Invicro LLC, and we are having some internal celebrations also. But we also wanted to share this with you, and we should have a drink on Invicro later at the bar downstairs. I hope you brought the champagne or something.
We'll be champagne if we want it.
So, and with this, I would like to pass on to Bill for the introductory talk. Thank you.
Thanks, Krishan. And thanks everyone for joining today, both in person and online. We are excited for a robust discussion around, as Krishan said, how users both internally at Invicro and externally at pharma and academic institutions are collaborating with each other to solve these challenging data informatics problems in an effort to answer these even more challenging biologic questions and advancement of their drug development programs. As a play on our 10-year anniversary, I thought it'd be interesting just to look at how we got here. Our marketing group did a nice job kind of recapturing a brief timeline history of Invicro, and we've gone through a lot of change, or especially over the last couple of years. From acquiring companies, merging with groups, specifically around the scientific and operational capabilities of all these teams. And, most recently with the acquisition by Invicro by KM, really wanted to just highlight on a couple comments on what that means moving forward for our users and all the scientists that we work with day to day. Konica, after acquiring both Ambry Genetics and Invicro, really set out to form a advanced precision medicine group that is combining world expertise across different bioinformatics, quantitative biomarker disciplines, from genomics, histopathology to radiology. And Invicro, as we look at how we support and fit into that platform, we really view ourselves as a key data analytics technology driver of how to best leverage the current practices in managing data, viewing and analyzing data, reporting on that data from a variety of different modality types, whether it be a variety of modalities within radiology. With Konica Minolta, we've been advancing into the histopathology space more, and how best we can collaborate with our Ambry colleagues, as well as, I think, the quantitative biomarkers at pharma that are producing other data sets that groups are wishing to merge and to analyze in a cross-modality setting to answer these complex questions. And so the tools and the people that we have organized across the enterprise will continue to form into a group that will be made available to you as customers from a deployed software standpoint, as well as the continued engagement we have with many of our customers in the pharma data analysis space for the contract research services we offer as well. So very exciting times here at Invicro, and we continue to make investments in our company to stay at the cutting edge of how we think about data handling, data processing, and general data analytics practices. A little more specifics on in vitro, we continue to stay as an organization laser focused on radiology and increasingly some histopathology-based assays and the data handling tasks, data processing tasks that come with that. What may have gotten hidden from the large acquisition from KM was us joining forces with the Imanova team and their scientific group and operational capabilities, their site capabilities, Core Clinical, who their team has a wide breadth of experience in late stage imaging core lab services, particularly on oncology, but other therapeutic areas. And so the challenges that our software team and the image analysis group here at Invicro kind of jointly face to help support the wide array of different types of data, again, from a data handling, data processing, data analytics standpoint, are only getting more complex. And we're looking forward to exploring how best to provide solutions, not just internally, but working with you as well, as we certainly appreciate and enjoy all the scientific collaboration with you as customers as we try to jointly build out these solutions to meet each other's needs. And how we envision the future building and supporting the wide array of these types of data sets is really continuing focus on the core software platforms that we develop in-house. But I think you'll hear a great theme today of how our software technologies can be incorporated into a wider picture, a larger footprint or ecosystem of different software tools for handling these different types of data sets that may fall within the radiology, histopathology category or complement to such. So you may be familiar with this diagram of how, IPACs currently and VivoQuant as a platform handle a variety of different preclinical and clinical data workflows, whether it's from a data sharing standpoint, data transfer standpoint, or from the nuts and bolts of viewing and analyzing the data. We continue to see IPACs as a core platform to manage and be a content management system for all things data related in imaging trial. And how we see the roadmap building out is where we can make investments on improving integrations to not just our own software, but other tools that groups are using so that IPACs continues to be a center hub of the wheel, so to speak, of how you think about managing your image data. With that being said, the current environment is very interesting in how the landscape is evolving of how groups are thinking about managing not just image data, but all types of data within pharma. And as you can maybe see from a few slides ago, even at Invicro and working with our Ambry colleagues and our KM colleagues of how can we best think of IPACs as a component of a larger data management strategy so that groups, when you have this single question of, can I look at a correlation of data output from a variety of quantitative biomarkers, that the technology and the infrastructure is not limiting to how you're accessing that data, how you're interacting with that data, how you're analyzing that data. And so that is really a core focus, and I think will be a theme of the year in how we work with groups, our current customers, our hopeful new customers we don't know yet, but hope to meet soon and even solve challenges internally. And I think what continues to be such an exciting time at Invicro is the symbiotic nature between the problems that are presented to us through our contract research business and the problems that we interact with our customers using our deployed versions of software. We really see that line getting more gray in how we think about supporting these data handling, data processing, data analytic challenges. And so with that, I think, it's a great way to set the tone for many of the themes you'll hear in these talks today. I'll try to shut up quickly because we have a great lineup of speakers that will be touching on a variety of these topics. But, I think it's important, especially at a day like today, to really make sure that from Invicro, you realize how much we thank our relationships and our collaborative time with you as our users, and also our own team. So there's a lot of people here today, hopefully, that users, customers get a chance to meet. You see myself, TJ, Rhonda, Melissa as really been the main interface to you in a lot of these projects, but there's a huge team behind us that helps support a lot of the product development, the features development, general application support, technical support. And so it really, I hope everyone gets an opportunity to interact with the entire team here that's from our side. And then again, we really should be thanking our users. You help drive our success. So with that, a little play on one of the first marketing campaigns we ever had at the company. If anyone caught it, I think it was back in 2013, but it was highlighted by a phrase, "See more." So let's help each other see more today, one voxel at a time.
Hey, hi everyone. My name is Rhonda Silva. I am a software services engineer at Invicro. It is my pleasure to introduce our next speaker, who is Glenn Hughes from Merck Pharmaceuticals. He is a fantastic business analyst who I've had the pleasure of working with very closely over the past year. And I'm sure he will give a great talk.
All right. Thank you, Rhonda, for the kind introduction. Hello, everyone. My name is Glenn Hughes. I'm a senior IT business analyst, and I hope you'll get some value out of what I'll be talking about today, and that is how Merck and Invicro have partnered to implement a global imaging management and archive system that's used by many different departments throughout the company. So we'll talk about how IPACS is used as a global imaging management archive solution at Merck. Merck is a large research-based organization, and as such, we've got many different departments that use lots of different instruments that generate a tremendous amount of content. So we'll talk about why IPACS was the optimal solution given the environment we work in. We'll also talk about the different workflows that are used in some of the departments at Merck, early and late stage clinical, archiving, and we'll talk about how we use the Sync client. And then we'll also talk about the process that Merck and Invicro use to get the true needs of our business users and understand that well. And by doing this, we're able to determine that our archive team needed a critical snapshot report. So we'll take a look at that as well. So this slide was actually developed by one of our internal colleagues. And the key point I want to make here is that, so I've worked in regulated content management for a number of years now. And typically it's been in with the document management, where you can put tools in place and you can train users so that you have templates and you can enter metadata in such a way that as it funnels downstream, whether it's a regulatory submission or whether it's trial master file, at the end point, you have something that you can kind of have more control of that process. But within this domain, whatever the instrument generates, that's what we have to work with. So it's all about the science, as it should be. But the point being is that there's no, at least I'm not aware of, there's no consortium where these vendors get together and say, we're going to create one standard way of creating output. So aside from instruments that generate output in the DICOM standard, a large percentage of the instruments generate output in a proprietary format. So that's the type of complexity that we had to deal with. So our challenges, and there was a few. First of all, as I just mentioned, we had many diverse image modalities. So the easy case was DICOM, where you have the header as part of the image. But then we have a lot of instruments that generate. So for example, you have the Bruker that generates output. You have your files, but then you also have your metadata that's stored in a separate file. And so you have to figure out where do you go find that information when you want to parse it and index it. And then we even have cases where, like the vector, for example, where you don't even have a header file. You just have image files that are generated. So you had to read what was implemented within a folder structure. And as you can see here, we've got many different departments. Some of them just needed a backup. They just needed a way to, they're generating all this content, and they need a way for them to go to point A and point B. But we have other departments that they needed a definitive source so they could link back to their electronic lab notebook. And because we have many instruments, we have lots of different vendors. So we had all those different types of integrations that take into consideration. And then there's the amount of content that's being generated. So for example, we have our largest size we identified was 35 gigabytes per scan. We have some DICOM studies that are well into the thousands per scan. And then our cumulative content as of today is over 180 terabytes and counting. And then we have a challenge with localized storage. And what I mean by that is that, so a lot of these departments tend to be siloed so that they may go out and procure an instrument, and then they get a server that comes with that instrument, and they plug it in, we start scanning. And so we're generating all these images, but then the images just start to pile up within that server, but then they have their own way of backing it up. It might be on a thumb drive, it might be, they might copy it to a network drive somewhere. So we didn't have that enterprise way of backing up our data that's getting generated, which puts the organization at risk. So our solution was to actually implement 2 instances of IPACS. The first one was called GMOS, which stands for Global Imaging Management Archive System. And that stores our regulated content, such as clinical and safety data. And then GMOS NRD, with the NRD standing for non-regulated data, and that stores basic research. So what GMOS and GMOS NRD give us is one single source of truth for storing images and associated data across the enterprise. And this gives a lot of benefits. So first of all, as we onboard these different areas, what we do is we tell them, define your taxonomy. We allow them to have some flexibility in the taxonomy they define, but once they define it, we need to stick to that. So it creates some level of standard within the repository. And then because we do that, now we can set security at different levels. We can set it at the department level. We can even set it down to the study level if the study needs to be more fixed within a certain group of users. Now we have roles within the system. So there's the archivist role, and we're going to talk about in a little bit what we mean by GLP archiving. You have an admin role, of course, but then you have regular users. So you have some users have managed rights, so they can control the life cycle of a study. And then but most of our users have read-only rights to the specific studies they have access to. And then finally, we have a solution that allows us to conform to 21 CFR Part 11 compliance in terms of logging information that occurs in the system and then reporting on that. So as you can see here, we now have, this gives a foundational platform for getting our images into the system. So we start by uploading them using the sync client, which we'll take a look at in more detail in a little bit. We can either do an automated way or do it manually. Once the images are in the system, we now have a repository where we can, for images that can be indexed, we can search on them. But even image types that aren't indexed, we minimally can at least search on the file name and a folder structure. And there's also metadata reports the system gives us as well. And then we have an area that performs GLP archiving, which again, as I mentioned, we'll talk about that in a little bit. And when a project is GLP archived, you can go back and restore different archive versions. So why did we choose IPACS? Well, first of all, it gives us that authoritative source. So now we've helped eliminate that problem of backing up. Now these different areas have a mechanism to get their content in the system, and so it's backed up on a regular basis. And as far as automation goes, we leverage the sync client. So what the sync client does is that as we have content that's generated in an instrument, or when an instrument gets it, or we get the content from the instrument to a network drive, we can set up a sync client so that it looks at what's generated in the source, compares it to what's in the target. And if anything's changed, it'll be able to detect that, and then it'll automatically sweep those changes. So we have that run overnight on an ongoing basis. Again, as I mentioned, we have the ability to do searches. So we can search on, so for images that can be indexed, we have a very rich set of metadata. I mean, DICOM's one example, but we have other image types that we're also able to parse, and we're able to map it over to DICOM metadata fields. So we can use that for searching as well. And it gives us that foundational imaging platform. So now we have the ability to manage the storage of images for all organizations within Merck. And then we've reduced our compliance risk. So now we can store and search and retrieve our information that's CFR 21, Part 11, and Part 58 compliant. So this is a really busy slide, but the key point I want to emphasize here is, there we go. As content is generated on our instruments, we can use a sync client to either pull it right off the instrument into the repository. Now, in some cases, we have either scientists do a manual transfer or we have some automations to get them on a network drive. So then what we'll do is we can, once it's on the network drive, the sync client can pull from that. But as you can see here, we can also reverse it back out. So for some reason, because this system doesn't integrate with the tools that are used to view the images themselves. So if we need to, for some reason, take a look at them, we'll need to export them back out into the network drive. And as you can see here, we've got sync clients installed at our West Point, our Boston, and our Singapore locations. And that's because what we found is that through our testing is that a distributed model works best for this, meaning that you want your sync client close to where the content's being generated. And also too, you can see here that we've called this out as being the Linux client, because what we found is that when we're trying to load massive amount of content from these different places funnel into the one repository, that Linux just works best. It's just the strongest solution for that. But at the same time, we also offer a Windows sync. If users, let's say they want to manage the automations on their own, that's fine. They can download and they can install that, and they can set up a schedule on their own if they like. Okay. And next, we'll talk about GLP archiving. So during the course of laboratory studies, certain types of information which is generated must meet certain criteria with regards to records retention. So first, it must be point in time preserved. So what that means is that at the end of the study, that content needs to be locked down so that it cannot be altered or deleted. But at the same time, it must be easily retrieved. So we've locked it down, but we need to be able to go back in the system and bring it back if necessary. And this is actually really important in cases where if we were to have the FDA come in and they needed to question the study, we need to have the ability to pull that information out very quickly. And it must be retained in accordance with the study's retention policy. So I've heard numbers ranging from 35 to 100 years. So this is clearly designed for long-term storage. So next I'm going to go over the process for archiving. So at the end of the study, first the scientist will, they'll log into GMOS, they'll identify the study, and they'll put the project in a closed state. And what a closed state means, it's not archived yet, but it is in a read-only state. So it means that anybody who has access to that project cannot alter any of the information in it. And then they submit a materials to be archived form. So that's a communication between the scientist and the archivist saying, this study is officially completed. It's now ready to be GLP archived. And then the archivist will log in the system and they will locate the study, and then they will electronically sign off on it. So they must provide their password and a reason for archiving, and that information is stored in the audit trail. Then the system will GLP lock the content, marking the project as being in a locked state. The archivist also has the ability to optionally print out a GLP report, which basically presents a summary of what was being archived. And then the archivist will then sign off on the materials of the archive form, basically saying this project now has officially been GLP archived. So the key point here is that we now have this mechanism to meet FDA CFR 21 Part 58 regulations. At the same time, we have a very easy way to set our projects in a state so that they cannot be changed, but they can also be easily retrieved. Now, in some rare instances, we want to unarchive a project, and the system does give us the ability to do this. So when we unarchive a project, typically we may have to make some small changes to it. When this happens, the system will automatically put the project in a version control state, meaning that anything you do at that point forward has to be tracked. And so once version control is in effect, and then we make some updates, and then we re-archive the project, then we always have the ability to go back and restore individually archived files or restore an entire archive version. So next I want to talk a little bit though about what the concept of version control in a GLP archive, because it might be a little bit different than what you're used to. So typically when we think of version control, it's simply working with a Microsoft Word document where each draft is a slightly different copy of one document. And so this is something that we typically see an EDMS system like Microsoft SharePoint, where the version control represents a life cycle of a single document. But in the world of GLP archiving, version 1 is all the files in that archive. Okay, and then if we have to unarchive it, whether we add, modify, or oops, that's not good. There we go. Anyways, whether we hit the wrong button there, whether we add, modify, or delete files, that will create the version 2. And so version 2 is everything that's in. So we don't copy everything from version 1 to version 2. Version 2 represents anything that was changed, and then also all the files in version 1. So in a GLP archive, a version represents the point in time lock for all the files in the entire project. Okay, now we're going to switch gears and go and talk about our clinical submission workflows. We have two. We have early and late stage. So as Merck works with our external partners, we typically receive images in the DICOM format. And we'll also receive ancillary files such as PDF and Excel. Now, during the course of these studies, Merck is obligated to retain only the minimally amount of personally identifiable information that's needed for research purposes. Any other PII information which exists, we can't have it. It needs to be scrubbed, or the file needs to be kicked out. So what we do is Merck has an internal QA/QC group that ensures that as these images come in, that no personally identifiable information which shouldn't be in the images isn't there. So how do we do this? Well, first, the external submitter will, so we use the, we have a solution called EXGMOS, where an external submitter or collaborator site will get a login screen. It's a very basic screen. It just allows them to log in, and they take their package of submissions and they send it over to us, and then that's it. It's pretty much done. And they just tag it with whatever the submission protocol is that that package of submissions is associated with. Once it comes on our side of the fence, it lands in a quarantine folder, and then the Merck scientists will get notified saying, hey, you've got a submission now to review. So they log in, they can run a metadata report, which allows them to see whether or not there's any PII information in those images. In the happy path, which should be always the case, the submission will get approved, and then the content will get moved to its final location. But if by chance there is any PII in there, the submission can be rejected. The system will then automatically purge it, and then a notification will go back to the external submitter saying, the submission had to be rejected. We also have a late stage clinical submission workflow. So we also work with collaborators that at the end of the study, they send us hardware that contains images on thumb drives, external hard drives, maybe DVDs or whatnot. And this stuff is building up in cabinets, and we need to get this out of there, and we need to get it into the repository. So, but in addition to that, the scientists want the ability to go in and they want to search on this information. They just don't want to dump it in there and leave it in there and just let it sit. So we have two different ways to also use the term tag this information. One is that we can configure it so that as we do a bulk upload, we can pick metadata fields, and they can put information like if it's a therapy or it's whatever particular type of information they want to put in that metadata field to tag it so they can go back and report on it later. But they can also wait until the uploads happen, it's in the system, and then they can select a particular patient and they can tag that as well. Now, when they do this, it facilitates querying and reporting. And additionally, since they're making updates to the DICOM images themselves, that information is audit trailed. So this is how our late stage clinical submission workflow works. It's kind of similar, except that, again, the images are already in-house, so they bulk upload them, and then they're stored in a temporary folder in GMOS. And then the images can be optionally bulk tagged. And again, they'll run a metadata report, so that not only allows them to check for PII information, but they can also make sure that the tagging they did was correct. And once everything looks good, then they perform a move to the final destination. And then once the images have been moved, then they can be queried at that point. Okay. So now I'm going to switch gears and talk about how Merck and Invicro have partnered to manage business requirements. So one of our responsibilities in IT is to make sure that we add value to our business stakeholders. and because that allows them to achieve good business outcomes, right? So since getting good business requirements are key to achieving those outcomes, I'm going to talk about how Merck and Invicro partner to meet the challenges that were involved with satisfying the user needs in our organization. So does any of this sound familiar? This requirements document does not simulate our user experience. We have a hard time reading this requirements document. Last minute changes to requirements came out of UAT. This resulted in rework and delays. We have a hard time seeing how these requirements tie together, and this requirements document does not present a visual of our system. Did that happen to anybody? Okay. So why does this happen? Well, I believe this happens because we follow, when we follow a more, I'll call traditional waterfall approach, where we sit down in meetings, we try to figure out what our stakeholders need, or they tell us what we need, we write it down, we create this massive functional spec, then we find a vendor or have a development team or have a vendor which adds enhancements to it. And then we install it in a validated environment, we perform tons of testing, and then finally the user sees it at UAT. Well, the problem with that is, first of all, that large text-based document, it's really hard for people to comprehend. And the other thing too is that requirements tends to be more of an evolutionary process. You know, instead of giving somebody something to read in a big bang way, it's just a lot easier to digest things in more bite-sized chunks. And even though a functional spec may be required from a validation standpoint, just in terms of eliciting requirements, it's not really a very good tool. And even though you might have a beautiful functional spec, if it doesn't meet the business need, it's not worth a whole lot. So what can we do about it? One thing we can do is, as we implement changes, get in front of the user as soon as possible. So if there's a requirement, implement it in a development or a sandbox environment, and get that in front of them as soon as they can. Whether you can do storyboards or whether you have a development team that gives you something that pretty much works, get that in front of them and then get their feedback early on in the process. And then once you've taken all these changes and you've strung them together, allow them to perform at the UAT. Instead of doing the UAT, even though we have to do a formal UAT at the end, do a UAT dry run at the beginning before you do any validated testing. And again, that gives them another opportunity to get feedback. And again, by doing this, it provides a level of hands-on interaction, reduces the risks of changes late in the project, and it just allows the project team to absorb changes much earlier on, which increases the chance that we can implement what they're asking for. So here's some of the benefits that, from a practical standpoint, of following this iterative approach. So what we did was we conducted a series of demos. So Invicro gave us a sandbox environment, version 2, plus some additional enhancements we had asked for. So we did a series of demos in a sandbox environment on this newly added functionality to ensure it met the business needs. We met with each one of the different areas. And in addition to that, again, as I mentioned, once we had that all built out, we did our informal UAT up front in the process prior to doing any validation. By doing this, we realized that the archive team needed a way to compare multiple versions of an archive project. So that came out of this process. And so to satisfy this need, Invicro developed a snapshot report, and then this paid dividends downstream when it really counted during UAT. So here you can see in the snapshot report, this allows you to, so basically you're comparing two versions of an archive project, let's say. So version 1 is going to be represented by date and timestamp, and then the current version is going to say current. And then you can include a history of the log changes. So when you run it, you see now everything that's changed between whether it's in the browser or in the web disk view, right? So that's precisely what the archive team needed. They needed a way to understand what changed between version 1 and version 2. Because again, as I mentioned, this is when version control is in place, and we need to track everything and know what changed. In addition to that, it gave us all, so instead of having to hunt through the audit trail, it gave this view of all the audited events that occurred. So you can see here, every time something got added to the system, where we had project state changes, where there was unlocked or the project was closed, that's captured here, or if a file was deleted. So by following this process, then different features could be shared as they became available. And that way we can incorporate feedback and review up front instead of waiting until development was finalized. And then so instead of asking users to try to validate requirements by interpreting pages and pages of text, they were able to relate to the verbiage to what they viewed in the system. So this helped reduce project risk because it made the validation phase much more predictable. And in addition to that, approving the requirement specification became a lot easier. So now instead of having to just understand pages of text, when we went through the functional spec, we could relay it back to something that they already saw. And as a result, UAT ran very smoothly. Any questions? Oh, thanks.
Excellent. Nice talk.
Sure.
Can you comment on the balance of a BA being very focused on understanding the technical components of the software or the app or whatever solution you're trying to deploy versus the focus on the documentation process and the transcription of just requirements back to the development team?
Okay. So as far as the technology is concerned, I guess I'm fortunate. I used to be a developer. So even though it's been a while, I at least have enough of a technical background that, like, when we communicate with your team, you know, we can have a conversation so that it's understandable enough. There might be a little bit more of a challenge for somebody who came to the business side. If that's the case, my recommendation would be just make sure you have a good SME. Whether it's a technical SME, just like I rely on SMEs from the business side as well too, because coming from that technology background, being in the IT side, even though I again, as I mentioned before, I have background in content management, sometimes you get in situations where you are in new departments and there's a totally new workflow you have to use. So having that SME available, whether it's a technical one or a business one's key, and you want to make sure you identify them, excuse me, as early on in the project as possible. Is that?
Thanks, nice talk. So I have a question about your two systems. I mean, at the application level, you have a regulated and non-regulated for development. But the backside, you have a multiple Apex server or just integrated in one?
Okay, where we have the sync clients?
Yes.
Yeah, that was to just show the concept that,
yeah.
If I don't remember if you saw, but at the bottom it had GMOS/GMOS NOD. It's just to show that
it's one server or multiple.
Oh, I see what you're saying. Yeah, no, it's each GMOS and GMOS NOD each have their own server.
It's separate.
Yes.
So then you have Singapore, West Point, and Boston. Also, is the individual or just the?
Yes, they are. Yes, we have individual sync clients, and so we have that distributed model where they're located at each particular location where the content's being generated.
I see. Thanks.
Sure.
So in your current practice, you only archive the original DICOM image, or you also archive the interim result during the analysis, generated from the analysis?
There's analyzed files that get uploaded too. I mean, it's up to the scientist, and that part of it I'm not as familiar with in terms of what it is. But you know, it's whatever they determine needs to be archived in that study. I guess there's certain rules behind that they need to follow. So, but I do know there is analysis information that gets uploaded.
And also, probably also sometimes some other documents need to be saved together with the image to help us understand. Like for example, if you do the functional MRI analysis, functional MRI imaging, and you need to know when they give the stimulus. And with that information, you can do the analysis. And just wondering if you have any standard procedure to when I put the archive the image in, and what kind of other information also needed to be saved in the same place.
The system will take anything. I mean, it's not restricted to, I mean, even though it's image centric, if they decide that they need to, if there's any Office files, PDF files, anything that needs to go into the system, then they can just put that in the location where it needs to be uploaded, and then it'll just get swept right along with everything else.
Yeah, I know the system can store everything, but just from the Merck standpoint of view, do you have any standard procedure about when they archive the image, what kind of information need to be included?
I'm not aware of one, no.
Can sort of answer that question. So we also have an electronic lab notebook. So we connect to, we reference the GMOS NRD location for the raw data, and all of the study parameters are contained in our lab notebooks.
Is that it? Okay. Thanks, everybody.